Just quick question on the actual virus. I know that you had spoke about, it parts of it starting into a lab that started in North Carolina and went to Wuhan, and that there was the use of this animal testing and animal pieces in this virus. But you're not saying it was necessarily created in a lab, but also you had said it couldn't have been created in 800 years. Can you talk about maybe where did this come from?
Yeah, so a best way to say that is natural evolution of a virus from the way it was described to us of this coronavirus from the bats jumping directly into humans, apart from the original SARS, which is the closest family member. That natural evolution of these families of viruses could be up to 800 years if it occurred at all. So what I said is it doesn't necessarily have to be engineered, as in we cut and paste, the scientists cut and paste and use recombinant technology and enzymes. We call it pseudotyping to make a new virus or things like that. That didn't necessarily have to happen, but the fact of culturing a virus in an animal cell line in a laboratory, that passing it through an animal cell line, the Vero E6 monkey kidney cell line, that the paper of the emergence of this virus that was published in Nature Medicine in 2015 by the investigators at Wuhan, North Carolina, Harvard, and Switzerland, I believe, that 2015 paper said the cell line originally came from Fort Detrick's USAMRIID, U.S. Army Research Institute of Infectious Disease in Frederick, Maryland. That's a biosafety level 4 facility. That cell line was sent to the Wuhan facility, and the manufacture, the growth of the coronaviruses in that cell line, again, which harbors not only relatives of HIV, but other gamma monkey retroviruses, simian virus 40, that were in the early polio vaccine. So those things could be picked up and carried in the preparation. So viruses, there's what we say in science is viruses rarely travel alone. Pathogens rarely travel alone. So one virus like this SARS coronavirus 2 is just an upper respiratory infection. So it does essentially nothing in most healthy people. But when you carry an HIV or components of an HIV and allow the gain of function, allow it now can infect white blood cells, both the innate and adaptive immune response. So your frontline defenders are being taken away from the battle against the coronavirus because they have to deal with the HIV. So you see those are called gain of function studies. So if this virus went by way of a laboratory, which that paper reports it did, because they cultured it in that cell line, it's not a natural evolution. And we don't know what all the components of the virus are, because you isolate the virus away from the other things that could have contributed to the pathogenesis and the disease causation. And clearly, the coronaviruses don't do some of the clinical endpoints and things that are being called COVID-19. Clearly, coronaviruses can't infect the brain. They don't have access to brain. Clearly, they don't, if they have HIV sequences, yes, you infect white blood cells. But what about those gamma retroviruses that are clearly in that cell line and that, in fact, infect every cell in the body through amino acid transporters, through phosphate transporters, which we knew the XMRVs, the mouse cancer-causing viruses we isolated from humans with these very diseases and with these very compromised immune system. The disease signature we published in 2011 is exact for that flame that's been attributed to SARS-CoV-2. And that's why I say this is fraud and a plague of corruption, because coronaviruses don't do that. So it has to be something else.
And the U.S. government gave $3.7 million to this Wuhan lab as well, that were experimenting with this same virus that originally came from North Carolina. Is that right?
That's correct. Well, not North Carolina, Fort Detrick.
Fort Detrick.
And so North Carolina was involved. So the cell line came from Fort Detrick. So the biosafety level is when you're a collaborator, the collaborators were all working with the tissues and the cell line. So I think it's pretty clear that Wuhan is where the animals, the bats were, where they were transferred to the cell line, the Vero monkey 6. But then it came, it was shared with the Harvard researchers, the Switzerland researchers, and the North Carolina researchers in those cell cultures were. And so this is another, how did it spread around the world? Well, cell culture is a way to spread viruses around the world unwittingly, unknowingly, certainly. But we know the risks, and those risks, and the we is virologists worldwide. And that's why these studies were illegal to be done, these so-called gain of function studies from 2014 through 17. Francis Collins said it was illegal to do this. So why is Tony Fauci's National Institute of Allergy and Infectious Disease, why is his agency funding investigators around the world much more than 3.7 million to do these studies?
And so why is he?
Because he has an agenda. He's part of the corrupt old boys network of our healthcare system. That these people have been in office or in their positions for 40 years. And so they're doing it to cover up their crime. This is their crimes of 40 years, and drive the agenda that once everybody's chipped and vaccinated, once everybody is in fear of losing everything if they can't go to work, or being fined or go to jail if you can't open your pub or your hair salon, people will give up and get the shot.